There are many reports that say that synaptic dysfunction causes or maybe is caused by these amyloid or tau plaques, and we haven't really measured that. And so I think to have a system that allows us to, let's say, take a disease model of Alzheimer's disease, for example, brain slices or an organoid model versus a healthy system, and we would be able to monitor the electrophysiology, so the electrical responses, simultaneously to the chemicals that are implicated, which are actually, I think, serotonin, dopamine, acetylcholine, and glutamate, then maybe we would be able to see differences in the two.
Hello everyone and welcome back to the Neuroscience and Beyond podcast. I'm Svilen, a PhD student in molecular and cellular neuroscience at the University Medical Center Göttingen in Germany. Together with my amazing team of graduate students, we are on a mission to bring the wonders of science, especially neuroscience, closer to you. Today, we have a truly fascinating episode lined up for you. We're exploring the world of small molecules called aptamers and their potential as biosensors in neuroscience. Our guest today is Nako Nakatsuka, an assistant professor and head of the Chemical Nanotechnology Lab at EPFL's NeuroX Institute in Switzerland. Professor Nakatsuka joins us to discuss the intricate science behind these tiny yet powerful molecules. In our conversation, we explore how aptamers can be engineered to detect changes in levels of neurotransmitters, the small molecules crucial for brain communication. We're going to delve into the structural properties of aptamers and why they hold so much promise for improving patient care, especially for conditions like Parkinson's disease. This episode does get a bit technical, but don't worry, we've added extra visual material in the video to help you grasp the concepts. For listeners on Spotify, please refer to the additional information in the description of this episode. Before we begin, I'd like to mention that this podcast is a separate activity from my position at the University Medical Center Göttingen in Germany. This podcast is supported by the International Max Planck Research School for Neurosciences, the Cluster of Excellence Multiscale Bioimaging, and the European Neuroscience Institute in Göttingen. If you'd like to support us, please subscribe to our channel and don't hesitate to drop us a comment under our videos. This episode, like others coming up, was recorded during the Neuroscience conference in Göttingen. And now, dear listeners, please enjoy the conversation with Professor Nako Nakatsuka. Professor Nakatsuka, thank you for coming to our podcast.
Thank you for having me.
We're still having Neuroscience up the hill in Göttingen, and we had the pleasure to invite some of the speakers. And you're one of the speakers, and you're giving a talk tomorrow, actually. And we are very, very happy to have you here and to talk about your research. Particularly, I'm very interested in learning more about the aptamers or other biosensors and how do you use these tools to study the brain. Saying that, can you tell us more about your research?
Sure. Thanks a lot, Svilen, for having me. And I guess to really summarize what we work on, we work with these DNA-based receptors called aptamers. They are unique in that they're designed to capture different small molecules, for example, neurotransmitters. So in my PhD work, when I first started, there were no good aptamers to capture neurotransmitters, but also not good antibodies, which are the more traditional bioreceptors. So we really needed to have an alternative strategy of how can we capture small molecules, which is not an easy feat because small molecules don't have a lot of surface area, not a lot of functional groups. You have molecules like dopamine versus norepinephrine, levodopa. They have very similar structural motifs, so differentiating them is very difficult. So the big advantage of aptamers is they can be designed in vitro, so also just in the system, in the lab, to be selective, to be able to differentiate the structurally similar molecules. And then we take these sequences and we integrate them inside of chemical biosensors. And so the mechanism is actually similar to how the brain works in that you have two neurons where you have an electrical signal that triggers the release of neurotransmitters in the synapse that bind to the receptors of the next neuron, and then there's an electrical signal. So we actually take the chemical binding with the aptamer receptor and convert that to an electrical signal by using biosensors that can transduce the signal from chemical to electrical. So that's the general theme. And while we're really interested in quantifying neurotransmitters for brain research, we can also use these aptamers for other applications like hormones, sugars, other kinds of small molecules that it can detect.
Wow, there is a lot that we can talk about right now. You dropped many, many, many terms. Let's start by defining first what is neurotransmitter on first place, and why is it important to know what neurotransmitter is released, where it is binding, and why cannot we approach this kind of study of the neurotransmitter with conventional tools? You already touched upon this topic, but can you elaborate a little bit on the basic kind of principles of this neurotransmission, for example?
Yeah, it's a very complex system, but I'll try to keep it as simplified as possible. And the general principle is that these molecules are signaling molecules, but the chemical version rather than the electrical signals. And they're implicated in a lot of basic brain function, whether it's memory, learning, consciousness, all of these different aspects that we hear about at the conference. But they're also implicated in disorders. So for neuropsychiatric disorders, neurodegenerative states, molecules like serotonin, dopamine, norepinephrine, GABA, glutamate that people have probably heard of are dysregulated. And so to give an example, for antidepressants that are given to patients who have depression, the mechanistic hypothesis is that it increases serotonin in the brain by binding to the proteins that allow the reuptake of serotonin. So again, that's a very simplified version of the mechanism, and I think there's a lot of more nuances and subtleties, but in general, that's how you can regulate a neurotransmitter and change a brain state from a dysregulated to a regulated way. But I think that because the brain is so complex and there are so many different neurotransmitters in different brain regions, by blocking one thing, it also affects many other molecules. So I think the limitation of conventional tools has been it's very difficult to monitor many molecules simultaneously. Also, the fact that they are structurally similar, they're very difficult to differentiate. So, of course, we're not the first group to have these neurochemical tools. There's many, for example, MRI can detect contrast agents that are either accumulated or single based upon a neurotransmitter that's present, but that's a labeled technique because you need a contrast agent. And MRI looks at very big picture, like at the millimeter or maybe centimeter scale. But then you can also go smaller to microdialysis, where you can put probes inside of brain regions and collect the brain fluid and analyze that liquid after. That's very powerful for monitoring multiple analytes simultaneously because you can use methods like mass spectrometry, where you can see a lot of different masses to charge ratios of different molecules. But it used to be at least very slow, and that you have to collect the liquid, then analyze and purify, etc. Now there has been many advances that have made it faster, but still it's not at the point of being real time, of really dynamically measuring what's happening. And then there's also, I think, a very popular technique is voltammetry, so fast scan cyclic voltammetry, where now you can implant an electrode directly inside the brain and you use redox chemistry, which is, for example, dopamine has these two OH or hydroxyl groups that are oxidized to the double bond O groups. That's what happens on the surface of the electrode. But the challenge of this is that also norepinephrine and levodopa have these same two OH groups, so you would see the same kind of oxidation peaks. So again, this method has improved over time in terms of using machine learning, new ways to differentiate these signals, but it's still challenging to detect 5 different molecules at the same time. And also, whenever you put something into the brain, there's this immune response that will start to coat the surface with glial cells or all these non-specific interference. So it's very challenging to do longer term recordings. And so I think every technique has all of these pros and cons. There's also the optical methods like the GPCR-based receptors that are tuned to capture different neurotransmitters and have an optical signal. There's the grab sensors, D-light sensor. There's so many new tools that are being developed. And I think every technique has its, again, advantages and disadvantages. Is it the spatial resolution, the temporal resolution, selectivity, sensitivity? And we are trying to fill some of those gaps by approaching some of the remaining challenges in these technologies.
So again, incredible description of all the other technologies that are out there. And as we know, some of them can be delivered to the humans via some, let's say, drugs or invasive delivery, or some of them are kind of a analysis tools to analyze samples from humans. When you say aptamers and biosensors in terms of your technique, how do we deliver these aptamers to the cells or to the brain, for example? Can we do this in the living animal, in humans? Can we do this in cell cultures or in tissue, like brain tissues? Can you tell us more about this aspect?
Sure. So I think in terms of biosensing for now, everything incorporates aptamers on surfaces. So you have this molecular receptor, you tether it to the surface of a sensor, whether it's a field effect transistor, which is what I worked on in my PhD, nanopipet sensors, which I designed in my postdoc time. So the readout can be diverse, but it's usually tethered. It's not just like injecting the aptamer into a body and seeing where it goes. That's potentially a future application, since aptamers have also been used for therapeutics, not just for diagnostics. But for now, for detection, we have to transduce that binding event somehow. So we have a surface underneath the aptamer. And so I think that the advantage is that we can also have different systems that we tackle, from in vitro, from neurons, where we can really start to go near synapses or to use disease models. You can go to ex vivo, from brain tissue slices or even organoid models. And then for the field effect transistor work, that's actually been miniaturized to be these neural probes, like the neuropixel, where you can really implant it in the brain of an awake mouse and do in vivo monitoring of molecules like serotonin. So I think that for every different system that you also want to monitor from, there's a different configuration that is optimal, and how small does it have to be, how sensitive does it have to be. So these are the questions that we have to ask depending on where it will be used.
Are you working on both methods, kind of the one in culture and the one in humans, for example, that is similar to the pixels, neuropixels, or you picked up one and you're working on that?
Yeah, so I guess there's been multiple phases where my PhD work really had this final application of in vivo, because one of my supervisor, Professor Ann Andrews, was working with these animal models where she really wanted to monitor serotonin in mice with depressive states or anxiety. So that was really the long-term goal, and we were able to get it to that point with the help of a PhD student after me who really miniaturized these probes to implant in the brain. But then when I moved for my postdoc, I wanted to ask a different question, which was really, can we get really close to synapses where these neurotransmitters are being released? Because there are very limited tools that have that spatial dimension, which is 20 to 50 nanometers, to really get close. So the questions are very different, but I guess I moved towards that in vitro ex vivo state. But now that it's been 5 years since I've been working on that system, now in my new position, we are also kind of going back to almost in vivo, because the institute that we're a part of is the NeuroX Institute. I think it's quite unique at EPFL, where there's so many different groups with diverse backgrounds, whether it's soft implantable electronics or spinal cord injury or MRI or computational. So I think we're now thinking about how can we support these groups with our chemistry as well. So for example, for these spinal cord implants, maybe there are chemical signals that can help the patients who are getting these implants, such as immune responses. And for these flexible implantables, what are some of the key chemicals that would be interesting to monitor? So I think we're again going back into that in vivo direction. So we're a little bit all over the place in terms of our interests.
Wow, that sounds amazing. All of these new technologies that are emerging, it's just remarkable. If we can go back a little bit to the mechanics of the technology that you're developing. So you have this kind of molecules, aptamers. Can you please elaborate on what they are and how do you measure actually the whatever you're measuring? For example, the neuropeptides.
This is my favorite part to explain because I think it's pretty cool. But you basically have these DNA sequences that in our case are designed to structure switch. So it really changes its morphology in 3D. So not all aptamers are structure switching. A lot of them have these lock and key type binding where you just have an aptamer, something clicks to it like antibodies. But in our case, they were designed in vitro to undergo a large conformational change. So DNA is very negatively charged because of its phosphodiester backbone. So if you have 40 bases in your DNA sequence, you have 40 negative charges. So now what we basically have is a DNA that has a certain structure, a molecule comes in, it changes its morphologies to really capture this analyte, which rearranges charge. So now as soon as you rearrange charge, you can start to transduce that at the surface of different materials, for example, semiconductors at the surface of field effect transistors, where now you can have two electrodes, a source and drain electrode, you have a semiconducting channel, and you have, if it's an N-type semiconductor, you have electrons that carry the charge from the source to the drain electrode. So when the aptamer moves its backbone, it changes how much electrons can actually pass through the semiconductor by changing the charge density at the surface. And so we can really record that as a change in current. Is the current increasing? Is it decreasing? And we now really know that depending on how the aptamer structure switches, whether it compresses or it elongates or something in between, we get a very different signal transduction in terms of currents increasing versus decreasing, for example.
It's remarkable. How do you ensure the specificity of this technique? Because as we know, the neurotransmitters, they have these precursors that are very, very similar to a neurotransmitter. How is the specificity ensured?
So I think what was unique about the aptamers that we isolated, and this is in collaboration with a group at Columbia University with Professor Milan Stojanovic and a very fantastic postdoc, Dr. Cassie Yang, and they really did this selection procedure, I think, in a way that was really designed for the final application. And I think that's where a lot of the mismatch happened beforehand, where there were groups isolating aptamers, so designing the sequences, and then there were maybe engineering groups that were using them on surfaces. But we know that because DNA is a molecule that is very sensitive to the environment, for example, the ionic content, temperature, pH, if the environment in which it was designed is different from the final application, it doesn't work a lot of the time. So where we really, I think, changed this is that we started with the final application of in vivo monitoring in the brain. So the aptamers were designed in artificial brain fluid that has the same ionic content. For example, it has magnesium and calcium, which oftentimes in PBS that you use in the lab does not have this. And then they also conducted counter selection, which was really key. So knowing that the dopamine aptamer needs to differentiate norepinephrine and L-dopa, they would have high concentrations of these analytes to off-compete any of the sequences that were cross-reactive. So it's not that we just found these selective sequences, it's that they were already designed to differentiate them within the selection procedure. But I think that at the end of the day, since these are molecules that are charged, they're negatively charged, the non-specific interactions are inevitable. Like you will always have positive molecules kind of interact with it, which is why I think we have a very easy and good control, which is you take the DNA sequence, but then you scramble the order of the DNA. So now it doesn't recognize the molecule, but it's still the same negative charge, approximate size. And so then you can do this thing called referencing, where you have a specific binding and then a non-specific binding element. And then if you see the difference between the two, you can take into account the non-specific binding, because we anticipate that the positively charged molecules that associate will be comparable, while one will go this big structure switching when the target binds and the other will not. So there's different ways, I think, where we've engineered the system to be able to differentiate these very small differences, especially in these very complex environments like the brain or in blood.
Wow. It's really, I'm mind blown by all of this. So when you say that you attach these aptamers on the micropipette, can we talk about dimensions? Because the synapses is very, very small structure, the synaptic cleft or the space between two pre and postsynaptic cells, two neurons is roughly 20 nanometers, right? And you really need to be so precise to go there and to capture this kind of the moment of neurotransmission.
Yes.
How do you position, how are these aptamers positioned in your micropipette and how big it is?
Sure. So again, we are not making claims we can go in the synapse. I think that's a whole nother challenge. But in terms of dimension, we have a capillary that's exactly like the patch pipette. So you can really hold it in your hand. You can put it in a patch electrophysiology system, but the tip is 10 nanometers. So actually approaching the size of the scap. But so the patch pipette is usually 1 micrometer, and we have really minimized this size to 10 nanometers. And then we modify that inside surface with the aptamers. And now the mechanism is a bit different from the transistors in that these nanopores basically can monitor ions that go in and out of these pores. And because the current that you measure through the pore is dependent on the pore size and the geometry of the pore and also the surface charge density inside the pore, again, when we change the density of charges based upon the aptamer structure switching, we change the way ions go through the pore. So that's the way in which we can measure changes. And so we can actually precisely position the nanopipettes using a method like scanning ion conductance microscopy. This is like atomic force microscopy, which I think people are more familiar with, where you can map the surface topography of surfaces by tapping on the surface. You can get a map. Scanning ion conductance microscopy does something similar, but instead of touching the surface, it uses ionic current as a feedback to, it's a phenomenon called ionic squeezing, where as it gets close to a surface, you see a decrease in the current, and by measuring the current, you know how far you are from the surface. And you can start to map the topography of cellular surfaces, for example, without touching it. So it's a less invasive way of mapping topography of systems. And this is a very well-established technique. And we were envisioning, no idea if it works yet, that we can replace the capillaries that are used for SICM, which are, again, similar. These capillaries exactly the same as what we use, but slightly larger pores. Now can we start to map the chemical topography after being able to isolate where to measure from? Because you can first map the surface and say, there's a synapse because it has nanoscale precision in terms of the topography. And then we can start to get close to that to measure. But of course, it's a challenge of, are we, in the synapse, a little outside of the synapse? I think that's going to be a big challenge of knowing where, but at least we know we can get close.
By the way, I just want to, there's a lot that we can clarify throughout the talk, but something not related was the patch pipette, which is a teeny tiny pipette that is put specially for electrophysiological experiment where people poke, so to say, neurons in order to measure currents or flux of ions so that they can study the physiology of certain nerve cells or brain cells. What I wanted to ask you is, have you studied the structure of these molecules in terms of, the structural biology aspect?
So another great question that I love to answer because that's a big part of our research. So a big part of the chemistry part of our research is really characterizing these molecular dynamics of how the aptamer captures a small molecule. And this really started as a question during my PhD work because we had a sensor for dopamine, we had a sensor for serotonin, we had this field effect transistor. One increased in terms of the current with increasing amounts of the molecule, and one decreased. And that was really strange considering the fact that the two molecules are very similar in structure, similar molecular mass, same positive +1 charge at the pH we're measuring in. And so it's like, okay, it has to be the DNA doing something in terms of such opposite dynamics. So we use a lot of spectroscopy, for example, circular dichroism spectroscopy, that allows you to monitor how the DNA folds in terms of the secondary structure. But these are solution-based techniques where everything is just floating around in solution. So it doesn't really mimic what happens when you tether it to a surface, where now it has less degrees of freedom. Now it's tethered. And so we have other methods like quartz crystal microbalance, where you can assemble molecules on a surface. And this method works in that it's a piezoelectric crystal, meaning that if you apply a voltage, it oscillates at a certain resonance frequency. You can assemble molecules, and then with higher mass, you have less frequent frequency changes. And then what's really cool is that you can also see compression versus elongation of the DNA backbone, because DNA is very hydrophilic, so it has a lot of water molecules attached to its backbone. So when it's compressed, you kind of get rid of water molecules from the surface, whereas when it's elongated, you have more water molecules attaching. So there's different ways to characterize it in these kind of indirect ways, such as height changes. But we also have a collaborator at IIT, Dr. Dennis Garoli, and his postdoc, Ali Douaki, has been able to do molecular dynamics simulations to finally visualize the structure switching. And I think that was really a moment that was super exciting, where we can finally see, oh, this is how the dopamine aptamer folds. This is how the serotonin aptamer elongates. This is a possible binding pocket of where the molecule gets caught inside the sequence. But of course, with these simulations, you get a variety of structures that are the most thermodynamically stable, likely structures. So we also have to do experiments to validate them. So a potential for that is FRET, Forster resonance energy transfer. You can put two dyes in different locations of the DNA backbone. If they're within a nanometer from each other, you have quenching. If they're further away, you see fluorescence. So that way you can also see kind of what part of the backbone is coming close or not. And these are experiments that you can correlate with what you see in the simulations to say, this is the most likely structure. So now I would say, have about 6 to 10 different ways in which we can characterize the aptamers. And it took a very long time even to know how and what to do. But I think it's a really exciting start, because now we have the tools. We can say, okay, we have a new sequence, but we know how to kind of go through the protocol to characterize it, to predict how it will behave inside the sensor. And perhaps there are some structural switching dynamics that are more optimal for certain sensors. These are the things that we want to figure out with our new kind of way of characterizing the aptamer target interactions.
So you explained this so good and so precise, but I just want to make the audience sure that all of these techniques are extremely hard to perform, especially FRET and all of the others, and of course the modeling. But have you tried also crystallography or?
Yes. Oh my gosh. Again.
Cryo-EM or?
Great question.
I'm talking because, we have in our lab this kind of we work on expansion microscopy. I don't know if you're familiar with the concept. And in one of the experiments, we were able to see the conformational change of calmodulin when it binds calcium. So probably this kind of aptamer is an interesting target to try and expand and see.
How big is this calmodulin?
It's very small. We achieve resolution that is one nanometer in below. So
we should talk. We should talk outside of this.
After the podcast, right? Okay.
Because I have a very sad story to tell about NMR and crystallography, which is something I worked on for 2 years at ETH. It was something I was very passionate about, of like seeing what's happening experimentally. And I had fantastic collaborators at ETH who really, believed in the project and let me work with them. And with Dr. Antoine Clary, we really did some NMR, where in one dimension you can see very clear peaks, where if you add serotonin, you see new peaks arriving. If you have the wrong molecule, you don't see it. Really beautiful technique. But then you have to move to 2-dimensional NMR in order to see the 3D structure. And unfortunately, you need low salt to have a very clean 2D structure, and aptamers don't function at low salt, or at least that's what we found out. So our aptamers actually need at least 30 to 50 millimolar of sodium chloride in order to fold in the way that it's supposed to. And for the NMR to have a clear data, it had to be under 20 millimolar. So again, that was the first, that sucks. So then we said, okay, we're going to try crystallography. So then I went to Professor Stephanie Jonas's lab at the ETH Hongerberg campus, and she was again very generous to be willing to let me try this. And what was so exciting was that I think we did like 20,000 screens, we got a crystal. Super exciting, because I know that that's the first very difficult step. We went to the synchrotron source, we shot X-rays at it, and we got a structure that was 4 angstrom resolution. And from my perspective, that's like really small, right? So I was like, yes, like we did it. But from a crystallographer's perspective, she was like, it's like looking at it underwater. It's like blurry. So it was really trying to optimize this crystal. Can we get it to lower resolution? I think it's just some inherent property of the molecule in the crystal that didn't allow us to get there. That was a really sad moment to kind of acknowledge that. Now we're still trying to see whether we could use the molecular dynamics simulations and to correlate that with the envelope of the 3D structure. But again, then it becomes a little wishy-washy again of, are we actually fitting something properly? So I think that was a very exciting but challenging story to recall, because it was like, oh, we're so close, but we're unable to do it. And with cryo-EM, I've talked to a lot of experts, but they think that because the DNA is such a flexible structure and also the binding behavior is reversible, that's what allows us to do real-time monitoring, that it captures a molecule, but then it releases it. So with this dynamic structure switching as well, it's probably very difficult to kind of visualize that in cryo-EM. And so we never tried, but no one was also willing to be like, yes, this will totally work. So I'm happy to hear about your technique afterwards.
We should really talk about it.
Yeah,
and
that would be
drop
crazy.
by the lab. I don't know, when are you leaving tomorrow?
When am I leaving? Right after Peter Dayan's talk.
Okay, so we can really think about that. Our institute is not that far away, so as anything else in Göttingen, by the way, reachable within 15 minutes by bike.
Amazing.
No commute needed.
Awesome.
So, by the way, all the techniques that you mentioned, this cryo-EM, EM stands for electron microscopy, NMR is nuclear magnetic resonance spectroscopy. Yeah. So, and the third one was the X-ray crystallography, right? All of these are techniques that are used in structural biology, and they basically try to find the structure of proteins and how are they shaped, and they study this and study the interaction between proteins and also other binding partners, etc. And these are, as well as the others, extremely hard techniques and very expensive tools, and hard to analyze the data, hard to achieve success. And you just told us about the science in the making, right? All the frustration, all the failure, but eventually now you know a lot more about your molecules than before, right?
Yes.
So this is a lovely process to hear from such an experienced person like you. So I wanted to ask you, so you mentioned you have the serotonin and the dopamine aptamers, right? Or sensors, let's put it that way. Was there any reason why do you have these two, apart from that your supervisor was working on serotonin, or was it like, okay, I want to see these kind of molecules because they're important in certain areas of the brain and they play a role in certain disease or certain behavior, if you wish? How did it happen?
Yeah, so I guess to be honest, it really started with my supervisor in my PhD wanting to make an in vivo serotonin sensor. Like that was the goal from the very beginning, which I very much respect, like very, focused. And then I think dopamine was really a comparative sensor. It wasn't that we had interest in doing in vivo dopamine sensing. It was, okay, we should at least have two sensors to compare the signals from. And so actually in that process, we also developed a glucose sensor and a lipid sensor, because at that point we were really interested in mechanism. And so glucose is a neutral molecule, and we wanted to demonstrate that it was the DNA structure switching that is the way in which we transduce the signal, not the target charge. And so it was really more of a mechanistic study at that point. How are these transistors working? Is the structure switching actually the way in which we're transducing the signal? Because again, I don't think we were the first people to work with aptamers and transistors, but I think that the literature was quite kind of cluttered in terms of what people were detecting, how they were detecting it. Sometimes people were detecting the same analytes with the same aptamers and seeing opposite sensor changes. And so it was kind of like, what is happening? Like, how are people detecting this? And now also having worked with different platforms, whether it's a indium oxide semiconducting surface versus a graphene surface versus a carbon nanotube surface, the signals are different. And so I think that the motivation started as this mechanistic aspect, but then of course I continued that with the nanopipet sensors, especially because we know how these aptamers work now, kind of. I had to still continue to characterize them. So I wanted to start with a working system. But then I think what was exciting was that I went and talked about it at conferences, and neuroscientists really reached out to me being like, I want that dopamine sensor, I want to use that serotonin sensor. And we really found these amazing collaborations where we could really make a difference in terms of quantifying those molecules in a system where it was previously not possible for a group that wanted to do a very specific measurement. So I think we really found our purpose with these sensors, but it was thanks to a lot of other groups that had very specific questions that they wanted to answer.
Before I ask my question about the glucose and the sugars, I'd like to ask you, do you plan to also develop aptamers for, let's say, glutamate or GABA, the most like famous neurotransmitter, if you wish?
So there's a science paper that just got published, I think last year in 2023. The lead author is Dr. Cassie Yang, who I talked about before from Columbia University, and it has identified, I think, almost 30 new sequences for neurotransmitters, amino acids. Like, it's an amazing library of different molecules. So we are now working on a GABA sensor. We're also working on a norepinephrine sensor. So we're really starting to expand the toolbox. And again, since we know how to characterize the aptamers, I think hopefully the sensor development goes smoothly. And we're really doing it based off of course, some questions that we have personally about going near synapses, what do we want to measure, but also some of our collaborators have requests of, I really want to monitor acetylcholine or norepinephrine. So yes, long story short, we are expanding the different kinds of molecules we can detect.
That is amazing. And as you know, guys out there, all these different neurotransmitters, they play different role. For example, GABA, it's more inhibitory. Actually, it is inhibitory neurotransmitter. Glutamate is excitatory. You have the dopamine that is important in the reward system, also in some disease-related aspects like Parkinson's. Serotonin and also other disease like depression and psychiatric diseases. Norepinephrine, acetylcholine, these are all important neurotransmitters that by understanding their function, we will understand our behavior, or let's put it that way. First the physiology, then how the things are working, and eventually our behavior. And of course, the ultimate goal is to try to treat some disease or some aspects of a certain disease, for example. Speaking of this, okay, before I go there, I wanted to ask you about the glucose. I'm studying the extracellular matrix, right, and the dynamics. And the molecules of the extracellular matrix in the brain, they are heavily glycosylated, right? They have these sugar chains and they have sulfated groups. Anyway, I don't want to go into detail. Do you think that we can detect something? Definitely you are not familiar with what I'm talking about, but this kind of glycosylation chains of different molecules, or because how does it work with the glucose? You briefly mentioned, but can you imagine expanding on other sugars, for example? Yeah.
Definitely. And I think that what you ask about is interesting because, for example, it's not a sugar system, but I can give an example with a peptide system in that there was an aptamer that was developed for phenylalanine. And we wanted to see whether this aptamer that can detect phenylalanine could detect phenylalanine in a peptide sequence. So now it's not a free amino acid, it's in a peptide backbone. And can we differentiate, for example, in the middle phenylalanine versus at the C or the N terminus? And we actually have a paper that just recently got published and that we can differentiate peptides that have these moieties versus kind of other structurally similar motifs like tryptophan or tyrosine that are replaced because the binding event is completely different. And so we see different kinetics depending on what peptides are going through the pore. And so I think that, again, I don't know the structure of the glycosylated sugar you're talking about, but the question would be, would you have to detect the whole thing, the whole structure, or would you be able to identify a certain kind of chain, a side arm? And would that be, I guess the question would be, what do you want to detect? Is it that there is something there, or is it that this molecule has to be captured and sequestered? So I think depending on the question, there are many ways in which we can design aptamers to serve in these different contexts.
Another off topic we just found. Great. Definitely we will ask you about this. So now is the time maybe we can talk about the applications of these aptamers. How do you apply them to study the brain, for example, apart from this obvious study of the neurotransmitters, dopamine, serotonin. Do you have any other kind of functional aspects or physiology aspects, health, disease?
So I think I can give two examples, which I think are quite easy to understand. The first would be Alzheimer's disease, in that there have been so many clinical trials that have, I think, mostly focused on acetylcholine-based receptors, or I think glutamatergic receptors. Don't quote me on that, I don't remember. But also the fact that there are these protein aggregates, right, like alpha beta or tau protein. But there has not really been a comprehensive study that monitors the neurotransmitters that have been shown to be implicated at the same time as this protein aggregation. So there are many reports that say that synaptic dysfunction causes, or maybe is caused, by these amyloid or tau plaques, and we haven't really measured that. And so I think to have a system that allows us to, let's say, take a disease model of Alzheimer's disease, for example, brain slices or an organoid model versus a healthy system, and we would be able to monitor the electrophysiology, so the electrical responses, simultaneously to the chemicals that are implicated, which are actually, I think, serotonin, dopamine, acetylcholine, and glutamate, then maybe we would be able to see differences in the two. And then in these models, of course, they're simplified, they're not real, but the real system is very difficult to study. Maybe it also gives us insights into why did the clinical trials fail? Are we targeting the wrong receptors or the wrong system? And it can also serve as drug screening platforms, where if you add a drug, now you are monitoring everything. Can you get to that healthy state when everything is kind of holistically looked at? So it's really providing tools to be able to kind of better understand what are the other systems that are playing an important part, in addition to the more conventionally studied protein aggregation mechanisms. So that's, I think, one area we're very interested in. And also we have access to these kinds of patient-derived samples or, animal model samples for Alzheimer's disease, which allows us to really do these kinds of measurements. And then I think the second one, which we're really excited about because it's something we're actually going to do, is for Parkinson's disease. And for this, it's actually a clinical study, in that in collaboration with the Radboud University Medical Center in the Netherlands, they have a cohort of about 300 patients who are going to have their brain fluid collected, which is a very painful procedure, through a spinal tap. So that's not something you can do all the time to check your brain dopamine levels, and their blood, which is much more accessible. And for Parkinson's patients, currently the only treatment for a lot of their motor symptoms, such as shaking, is levodopa, which is the dopamine precursor. And levodopa is orally taken, and the hope is that it goes through the blood-brain barrier and makes more dopamine. But of course, levodopa will also circulate in the periphery first, and there are enzymes in the gut and the blood that can convert it to dopamine. And once it becomes dopamine, it can't cross the blood-brain barrier, so it's not really doing its purpose. And a lot of patients have a lot of side effects because it could be the blood dopamine is toxic, so it's actually not going through the blood-brain barrier. You have a lot more dopamine in the system. So the patients actually take a co-administered drug called carboxydopa, which then blocks the enzymes in the gut and the periphery. But again, that's so dependent on the metabolism. So if the patient ate something that morning, did some exercise, depending on their age, their health, their gender, whatever it is, it's very different, but it's not a personalized treatment. So in this study, we're trying to see whether there's a correlation between L-DOPA drug dosage versus blood dopamine levels versus brain dopamine levels. If there's a correlation, then blood dopamine would be a very good feedback system for patients, where I think it's a very frustrating disease because there's only one treatment and it works sometimes, other times it doesn't. For some patients, it doesn't work at all. And we have to figure out why is that. So I think what's really exciting about this project is that I always thought that we're very much focused on fundamentals still because there is so much that is unknown and it's such a black box. But I think we're finding ways to actually support patients without, claiming we're going to solve the disease, but to improve the quality of life or to give them feedback for their drug dosing in these kinds of systems where we can quantify molecules that could be important.
Very exciting. And I will mention again that we also study Parkinson with our one microscopy technique, another topic. But yeah, L-DOPA is some drug or precursor, right, that works to some extent, and then at some point it stops working, even in the people that were treated well for a certain time. And there is a next stage that could be the deep brain stimulation, where actually people implant, doctors implant this kind of electrodes into the brain, into the deep brain nuclei, and stimulate the area of the brain where the neurons are degenerated. And these neurons that produce dopamine, another molecule that you're studying, right? And yeah, it's very interesting exactly to answer these questions that you just mentioned about why is it working in one person and not in the other one. And it's very, very important to increase the health quality, the life quality of a patient, because it's a horrible condition and we still don't know how to treat it. We don't know the exact origin or cause of this disease. Also in Alzheimer, the same case. We have the plaque amyloid beta and the tau tangles, which are two, let's say, protein aggregates that are a marker of this disease. But we don't know why are they there or what causes their aggregation, let's put it that way, and when is it occurring, which is even more crucial because in many cases, our failure is to diagnose the people, right? And most of them are post-mortem diagnosed with these diseases. Therefore, it's incredibly important to be able to develop sensors that can detect things, that can improve the health quality, the life of patients, right? So I'm really fingers crossed that you achieve a lot of success with the biosensors in this regard, and I hope that you develop also sensors for other diseases, not only for the neurodegeneration. What about the neuropsychiatric disorders? Serotonin is involved, right? Do you work in this regard also, or not yet?
So not specifically. I think that, again, we are not really experts in these diseases, so it really depends on the clinical partners that we have, the people who can really support us in this. And I think the reason why we actually started focusing on Parkinson's disease was also because we started talking to patients and caregivers who actually have a lot of information, which I think is really fascinating. One of the new aspects that I also learned about is the sex differences in Parkinson's disease, where there is a whole community of women who are saying that the drug dosage completely relies on the menstruation cycle. And they know this because they feel it, and they can really keep track of these things. But when you tell a doctor, they don't know what to do about that. So then we can also start thinking about maybe tracking hormones at the same time, like estrogen, progesterone, testosterone, can also maybe tell us whether the levodopa is dysregulating or altering the way in which these hormones are interacting as well. At the end of the day, I think it's such a complex system because there's so many connections. One thing can affect a totally another system. But I think that's why we're really kind of following the trail of what patients are telling us they want to learn or need. And I think that that's in the area of Parkinson's disease for now. But of course, we're not saying that we'll never work on in other kinds of psychiatric diseases. But for now, this is our focus for the next few years.
Yeah. So you have already established something and you want to finish it, and then probably expand depending on how things are going. Of course, once again, all of these techniques and method developments are extremely, extremely hard. And really, I deeply appreciate the work that you're doing.
Thank you.
So I want to switch the topic a little bit and go more, private sphere, right? I wanted to ask you, was there some kind of a drive for you when you were, let's say, undergrad student to study this kind of things, or how did you end up actually studying aptamers?
Yeah. I'll try to keep it short because it's quite a long, up and down journey. But I went to the United States at 17. I left Japan by myself. And I went to a university called Fordham in New York, and I honestly didn't really know what I wanted to do. I really liked chemistry as a child as well. Like I just thought it was cool, but I never really had this drive to be a professor or anything. I was just like, oh, chemistry is fun. But I also said, maybe I'll go to art school. Like I also like to paint. Like who knows? But my very strict Japanese mother was like, you're going to go do science if you're going to go to the US. And so off I went. And I think I was very demotivated by the courses because it was not very applicable. Like it was, you can do organic chemistry and learn all the mechanisms. And I was like, who cares? Like, what is this actually doing to help society or anything? So I was really kind of down for a while. And then I really met someone who completely, I think, changed my life, which was Dr. Banerjee, Ipsita Banerjee at Fordham. She was organic chemistry lab instructor at the time, and she was my teacher. And I was complaining to her that I have no purpose, like I don't enjoy the things I'm learning. And she allowed me to work in her lab for the summer. I was, I think, in my second year summer. And it was a whole new world of research, which I didn't really know about before. And she worked with tissue engineering, and she really motivated it in a way that made sense to me because I was also an athlete. I was running competitively. I just broke my ankle. I was on crutches. And she was like, what if you could, inject something that would help to regenerate the tendon that you tore and you wouldn't have to get surgery? And I was like, that sounds great. Like, let's do it. So that was really the kind of system she was building: cartilage regeneration, tendon regeneration. And so I was also very passionate about it because I had those injuries, knew what it was like to go through that. And it was really the first time I was like, oh, this is very applicable. I can see the purpose of doing this. It was pretty tough because we had to commute to Queens College to do all the research, 2 hours away from the Bronx. And it was really like I'd go to running practice, take classes until 5 p.m., 2 hours to Queens College, research until 2 a.m. and come back. So it was not a maintainable schedule, but I think it was really like, oh, this is finally something I'm passionate enough that I don't care how much I work. Like, this is what I want to do. I don't really care about classes. I even had a B in some of my chemistry classes. And then she was the one that said, if this is something you enjoy, why don't you go and do a PhD? And again, I never really thought I would do a PhD, but I said, I like research. This is something cool. I applied to a lot of different programs in the United States. And I think at interviews, it was really funny because nobody knew Fordham because it's not known for research. But I had a couple of first author papers as an undergrad, thanks to her mentorship and supervision, which I think allowed me to be competitive as a PhD student. And again, I met Paul Weiss and Ann Andrews at UCLA, and they offered this project, which was kind of this very big picture. We want to measure serotonin in the brain. We're not at the point where there's a sensor or anything, but, would you be up for the challenge to make this happen? I was like, that's super cool. Yeah, why not? So I think it wasn't that I thought I would work in neuroscience, and it wasn't that I thought that I would work with aptamers, but it really came from wanting to solve a problem that seemed like a really cool challenge. But then along the way, I got very passionate about it because I think it's very fascinating. It's interesting. I really wanted to understand how these aptamers were interacting, which really kind of kept driving my curiosity and my postdoc. And so I've just kind of been following the things that are interesting and make me passionate. But it was never really like a drawn out path for me from the start.
What a great story. And now you're in Switzerland,
yes.
Right? And Switzerland is famous for a lot of biotech companies, especially in Zurich, in Basel. I don't know about Lausanne probably as well. What makes you still stay in academia and not leave?
That's a good question. And I've also looked into industry at some point. It's never that I was on this, one-track academic lifestyle, but I think what always drew me back is that I really like students and I like to teach. And I think that's what makes academia special. That's kind of the extra aspect. I also get to lead a team in anything I want to work on. So that also flexibility and freedom, I think is really exciting. But I always told people who are also into academia that it's also not what people think it is to some extent. Like the higher up you go, the less you're in the lab. Now I get to be a little more back in the lab since we're new and getting started up. But I think the more writing you do in terms of grant writing, which I actually enjoy, like I like writing scientific work and I like to brainstorm things on paper. I like to teach. So I think all the components of it that sometimes I feel like people don't realize that that's the full package of academia, a lot of management, a lot of people interactions, a lot of communicating science. If you don't enjoy that, it's probably not for you. And I think going to ETH was, again, a very fantastic, eye-opening experience for me because I came from a chemistry department at UCLA. And I think the mentality was much more like, we do academia, like we care about fundamental chemistry and like industry bad a little bit, to some extent. And going to ETH where there was the lab I was in, the Laboratory of Biosensors and Bioelectronics by Professor János Vörös, there were so many spin-out companies all the time in the lab. And then I also got to see that they were like an academic group, they're working in the lab, they're trying to solve a problem, I guess on slightly different time scales from us, but I see how exciting that can be. So I don't think there is, anything wrong with either path. So I would have also been excited to work in a startup company working on an important problem. But now I also get to mentor a lot of students, which is something I enjoy. So I'm happy to be in the position I am.
Who knows? Maybe if you continue at this pace, probably you can spin off your own company, right?
Maybe we're thinking about it. Yeah.
Nobody knows. Professor Nakatsuka, it's been a pleasure to talk to you and hear more about your story and the person who you are, right? And about your research. It's time to bring you back to Neuroscience so that you can enjoy the conference and the dinner and the party. Once again, thank you very much for your time.
Thank you so much, Phelan. It was really nice to talk to you.